Yes, well, it is indeed my pleasure to be introducing today a colleague and friend, Dr. Maria Pachiadaki. And she comes to us from Woods Hole at the moment, but she's traveled the world. She started with her BS degree in Athens, Greece, in biology, got her master's in Athens in biological oceanography, and her PhD at the University of Crete in Greece in chemistry. All right.
Long story, if people are interested about
Over lunch.
Yes, over lunch.
She did a postdoc in Kaiserslautern, Germany, and then came to Woods Hole as a postdoc, which is when we met. She was working with Dr. Jim Edgecombe. She did another postdoc at the Bigelow Laboratory for Ocean Sciences. She is an expert on microbial processes in the dark ocean and has published extensively on it. She's also the recipient of a Simon's Early Career Young Investigator Award in Microbial Ecology. Kudos. And I'll leave it at that.
Thank you so much, Gordon, and thank you for, it's an honor to be here. As Gordon mentioned, we have been collaborating for a number of years now, and it's always a pleasure to visit like this part of the country. And like I'm specifically glad to communicate my science to you all. To students, and I'm a little bit facing like a small medical issue. And I'm a little bit more not as enthusiastic as usually I am, but hopefully science will give me back the motivation. I will start by saying that we, like my institution, sits on an ancestral land of the Wabanong people, and I would like to start this talk by acknowledging the people that they have lived here long before I went there, and the Western civilization actually went there. But I will start my talk by introducing you to my favorite form of life, which is the microbes. And I love microbes for several reasons, but one of them has to do with the fact that they are the oldest form of life. So this is a schematic of the time of that the universe is expanding, starting with the Big Bang there. So that is the 3.7 billion years ago. Earth is believed to declare its independence 4.5 years ago, and then we believe that the life has started around 3.8 billion years ago. So the microbes are in this planet 1/4 of the universe existence, which blows my mind. And we actually say the way that life is on Earth. And one of the facts that you might know is that cyanobacteria, with the production of oxygen, led to the oxygenation of both the atmosphere and the sea. But they are not actually saved the Earth when they start existing, but they are saving the Earth still now. So from what we know from the marine realm is that microorganisms actually mediate all the biogeochemical cycles. Of course, they are involved in carbon cycling, but of course, sulfur cycling, nitrogen, and some of the gases that they are like in climate active. But they look like that. So this is how like microbes look when they are stained in a way that their genomic material is stained. And of course, you have to use the microscope because, as their prefix suggests, they are tiny and they are indistinguishable. So actually, in this drop of seawater, they might be more than like 10 phyla present there. And when we say phyla in the microbial world, is like thinking an ecosystem, like all chordates, it's one phylum, sponges is another. So it's as having an entire like ecosystem all together there. And they are also very abundant. So the estimation for the euphotic ocean is around 1 million cells. in a very tiny cube of seawater. And although the abundance declines with depth, like very rapidly, and they actually encode for a lot of like genomic information as well. So in this small tiny cube of seawater, there is roughly 1 terabyte of genomic information. And the entire ocean has 10 to the 29th microbial cells, according to estimations, which is believed to be more than the stars of the universe. So again, another fascinating fact about like microbes. The problem that we have when studying the microbial inhabitants of the ocean is that most of the marine lineages escape cultivation. So we cannot directly interrogate them for what they are doing. We have to use different ways to do that. And this is a little bit an overview of how we have been working around this problem. So usually this is, let's say like a specific water layer that we want to study that has like a huge amount and diversity of microorganisms. And what we do, or my lab tries to do, is have an ecosystem approach. So try different like ways, approaches to do that. Of course, during the sampling, we do the physical measurements. We also do some chemical analysis of what is there. For me, as the oxygen, as you hear, is very important, and some of other gases, of course, the nutrients. I work with people that they were doing these rate measurements that I will talk a little bit more about. But I mostly do this part here. And this part here is trying to identify all the individual members. And there are several ways that you can do that. One of them is to filter all of the community members and then extract the material and reconstruct who is there. The other one is to do physical separation of the cells before inquire them about their IP and the genomic potential. And I work with people that they do other parts of that have to do with the with actually the role of virums, of the virus fraction. But as I'm a proxy of activity, I also do metatranscriptomics in order to be able to combine them. And the overall goal of that is to be able to go from who is there, that the taxonomy says that we can do through classification and phylogeny, combine that with the data from activity measurements to do the metabolic modeling, and then later on with machine learning approaches to define like metabolic strategies and construct ecological models, be able to do forecasting of the ecosystem. I am particularly interested in the dark ocean, and I have highlighted here some of the facts about its significance. It is the vast majority of the ocean volumes. It actually holds 3/4 of the pelagic marine prokaryotic biomass, and actually it's more biomass than the hydrotrophic levels. It is physico-chemically diverse, but it also has feedback to the climate that sometimes it's not it's not obvious because it seems that it's isolated from the atmosphere. And my lab motivations is to reveal the identity, evolution, and function of these ocean microorganisms. And what I'm mostly focusing my efforts on is to go from the community level to see how the community behaves to single cell, because as we, like microbes, have a lot of heterogeneity that we have only started to understand. And then like see those levels into the global ecosystem processes. And among all the microorganisms and the different lifestyles they have, my heart is closer to chemoautotrophy, and this is a mouthful, but I will try to explain it by chopping down the different components. So for life to exist, it needs actually to build bigger organic molecules from smaller ones. And we do that by consuming food that it's already an organic compound, but initially the source of carbon to go up the biomass needs to be an inorganic one. And those chemoautotropes, they have the autotrophic part, so they can feed themselves. They don't consume organics. They fix bicarbonate in most cases. But this is a process that costs a lot of energy. So in order to get energy from that, they use a chemical compounds that they are from organic origin as their electron donors. And I will explain a little bit what I mean about all that. But they have significance in the in the mesopelagic and below, because they provide like fresh organic matter there. And of course, they capture CO2, so they mitigate some of the climate impacts of increased emissions. And then, as I said, they also participate in important geochemical transformations. In all cases, though, this inorganic source of the material in the ocean, with an exception of hydrothermal vents, is coming up. It starts from the euphotic ocean. So in this talk, I will focus mostly of who and where there, and how we start addressing the how. Again, the dark ocean is interconnected with this surface, and what is happening is that carbon and nitrogen going into the system through photosynthesis. After this euphotic zone, when the light penetrates, there is a lot of primary production there. Those organisms will die and they will start sinking. And this dissolved and particulate organic matter will be eaten by the heterotrophic bacteria. They will consume oxygen in the process. And this organic matter degradation will release one of these compounds that they can serve as a substrate for the chemoautotropes. So there are two major groups in the majority of the ocean. The first one does the transformation, the oxidation of ammonia to nitrite. So this is the ammonia oxidizing. They are usually archaea. And then there is a second group, nitrite oxidizing bacteria, that they will convert the nitrogen to nitrate. And in the majority of the ocean, there is no one that will use this substance. So the majority of the ocean, actually, the dark ocean, has a lot of this compound. Until recently, people believe because of this group of ammonia oxidizing archaea was very abundant, that those were the most the most important chemoautotrophs in the ocean. But during my PhD in the Bigelow, we actually saw that the second group, the nitrite oxidizing bacteria, play a bigger role. And I won't explain those graphs in detail because I'll save the time to explain similar results in my ongoing work. But here shows that this group actually is involved for a high percentage of the inorganic carbon that is fixed. And when we do when we calculate the relative abundance in the ocean, we see that they are not in the epipelagic ocean, but the different groups can be either in the deeper water masses or in the mesopelagic, and there are some that they seem to be in some oxygen-depleted systems. But to go back to the introduction, I was when this process happens, when the organic material is degraded and oxygen is used, that draws the oxygen that it's dissolved in the water column down. And in specific areas in the ocean, either due to circulation barriers or to other factors, oxygenated water from the bottom cannot come in order to to oxygenate this middle part. So that draws down the oxygen to undetectable levels, and that actually makes the bacteria that they are there to use alternative ways to breathe. And alternative ways to breathe, meaning that they are not using oxygen that most of life on this earth does, but it uses these oxidized nitrogen compounds to dump their electrons. So at the end, this denitrification process leads to the release of nitrogen from the ocean that can have a feedback to primary productivity if bioavailable nitrogen is removed. But it can also lead to the release of this gas, the nitrous oxide, that it's actually 300% more potent than CO2. And that might, again, have a big impact on the climate. There is another process that's happening in this oxygen-depleted waters that I want to talk about, the anaerobic ammonia oxidation, and this also is mediated by a chemoautotrophy. And the process that it's less known is the processes that have to do with the sulfur cycling and how sulfur cycling might play a role in this carbon capture. So the organic material also has sulfide compounds, and some of the bacteria that they live, especially in these oxygen-depleted zones, they actually oxidize this sulfur. And it's believed that other bacteria reduce that back to sulfide, and there is a cryptic sulfur cycle that it's not very well understood. And to wrap up this introductory part, I want to highlight the oxygen depletion in the world's ocean. So everything that has blue colors there in the map means that in the water column somewhere, and it's not the entire water column, it's only layers in the middle that they are very low. Blue colors mean that it's not enough oxygen for the eukaryotes to breathe. So it's volume-wise, it's not much, but as area, it's quite important, and those areas they are believed to be expanding as the climate is going up. So starting with the a part of the results and what we have been doing so far, my lab has been doing so far. I will start you by introducing this Gort's dart that I've been referring to, and this is a reference genome database from the Dark Ocean. This is an effort led by my former postdoctoral supervisor, Ramona Stepanauskas at Bigelow, but we are still very good collaborators, so I get to deep into those still unpublished data in order to figure out the distribution and ecology of the chemoautotrophes. And those actually sampled 24 locations along those water columns. So there are 58 water samples from different depths. And as you see here, from all the oceans and a few enclosed seas. And the important ones for this talk is the Black Sea and the Baltic Sea here. Those due to circulation issues, they are actually the exhibit this anoxia that I talked about. And how the pipeline works for this in order to create these databases is that from each one of these samples, through a fluorescent activated sorting machine, those cells go to individual wells. So each well has one cell, and then we sequence those cells individually. We don't get the entire genome. We get partial genomes, but we still they're still very informative of what their capabilities are. So from all this effort, we actually sequenced 16,000 cells, and together with the sorting and the scatter parameters of the sorter, we get the estimation for the cell sizes. And then after the cleanup and all our subsequent pipeline, we ended up with a little bit less than 10,000 of cells. And we run them against databases that can tell us who is there, the taxonomy. And then for some of the results I'm going to show you, we estimate their distribution and variation. So who is where, and then we recover the metabolic potential. But I also have similar data sets from the two cruises that I have been leading. These are cruises in the two major oxygen deficient systems, Eastern Tropical North and Eastern Tropical South Pacific. Gordon Taylor and his team were a part of that, and we had a lot of fun sampling those areas. So we got around 2,000 cells from those areas as well, but from those ones, we also have more data. We have whole community metagenomes. We're working on getting the metatranscriptomes, and Gordon and other teams were doing the rate measurements. When I'm identifying chemoautotrophies, I'm looking actually for two, I need to have two different pathways to be present. One is the fixation pathways, and I don't want to go to details which genes I'm using. I'm happy to talk to people about that because they are at least 6 major of them with variations, that they have different requirements. But of course, which is the energy source, which is the chemical compound that serves as the electron donor. So ammonium, as I said, and nitride are considered to be the most widespread, but then I was looking for sulfide and hydrogen and other things. Together with the genomic potential, I'm also analyzing the GC content and the genome size and cell size of those genomes. And I would start by going into the results. So this group, Nitrosospirales, and for people that they have been in the field a little bit a few years ago, there has been a lot of renaming in those taxonomic in the taxonomy of bacteria. They used to be called Marine Group 1 or Chemoautotrophic. And apparently, yeah, we have already established that they're one of the most abundant groups in the ocean. And actually, we can see here, these are from all the samples, and I have categorized them in which ocean layer they are. So mesopelagic is from 200 meters until 1,000, bathypelagic 1,000 to 400. Abysso from 400, I think up to 6, and then Hadal. So the relative abundance is among the cells that we have sorted. It's a rough count. How many were belonging into this group? So of course, as we expected, the mesopelagic has the highest abundance of them, around close to 30%. It drops a little bit as we go deeper, but generally they are the dominant one in the data sets. Those are the oxygen-depleted systems. So Black Sea, Baltic Sea, South Pacific, and North Pacific. And they seem not to be as big of players in those areas. This is a group that requires oxygen in order to do the ammonia oxidation, actually. So the deepest part or the core of this oxygen-depleted system might not be a suitable environment for them. And we found the genes that they are responsible for the carbon fixation, and the gene that is responsible for the chemical transformation. Those go hand in hand, as I have shown you in the previous slides, with the second group that does the nitrite oxidation. And nitrospinalis is the group actually that is that will do the second part. So they will do the nitrite to nitrate. These are numerically less abundant. They are roughly 1/10. There is an exception here. 1/10 of the archaea, but they are bigger cells. And the fact that they are using different parts of the carbon fixation might make them more efficient. So again, there is still the results I have published say that as carbon that is fixed, this group might be more important, but the jury is still out. There are other groups that this one is the most important. But I will focus a little bit more in this nitrite oxidizers. And before I show you some of the insights that we get from their evolutionary relationships, I will briefly explain what the tree inform us for. So the way that we're able to categorize cells that they are not indistinguishable is by calculating how distant one genome is from another, and then we present them in a tree. This is a circular format, but it can be in other formats. That will actually the tree distance. So the distance of these branches here tell us how far apart phylogenetically different genomes are. So those here, the tips actually, they called trees, and those are the different genomes that we have examined. And sometimes we refer to clades when groups of we group genomes together. And this looks ugly, but we can actually overlay other type of information that we get. And the first information we overlay has to do with the taxonomy that we get from individual independent tools. So here I have the big colors in those clades are the two different phyla of nitrite oxidizers that they exist in the marine realm. And as you see, the vast majority is the nitrospinus that I just mentioned before. So I won't talk about the yellow group here. And those actually, not all of them are the same. So those colors here represent different genera that they exist there. So genera are distant relatives, but we identify individual species. So from those 190 genomes of nitrospinus, we found that they belong to 6 different genuses, and then 31 representative species. If we overlay other type of information, and this information we overlay here in these circles, has to do from the origin. So all the bluish colors in those tips mean that those genomes are coming actually from parts of the ocean that they have oxygen, and everything in red from the oxygen-depleted system. And this oxygen-depleted system has slightly different color because they are, from what we saw from the analysis, they are not they don't have similar members. And one of the things that first struck me is those clades here. And as you see, these clades have very long branches. And these very long branches means that they are phylogenetically very far from, and those with stars are the cultural representatives, that they are very far from what we knew that existed so far. And if you see for the colors there, one seems to be coming only from the ETSP, and the other one only from from ETMP. So from these anoxic systems, we recovered like genera that they seem to be endemic, but we also saw that within the same genus here, this big one that it's called Marginitomatota, there are like subgroups, and those are different species. That some seem to be endemic. They're coming only from the oxygen-depleted system, and other ones that they are coming like mostly from the normoxic ocean. And my initial question was, we have a lot of genomes, but it's still a pretty low number. So the total of the database is 190. So do we see this separation just because our numbers are low, or there are actually, we have indication that this is a global trend? And in order to do that, we actually used, and this is a complex figure, but I will guide you through that. We used metagenomes that they are in publicly available databases. You might have heard of the Tara Ocean metagenomes, of geotraces, and other systems. And all our individual species are here, and in the background I have colored the like in which genus they are coming from. And each one of the very thin rows here, it's one of the metagenomes that we calculate, the relative, represents, the color here represents the relative abundance in each one of these samples. And here we have actually graphed some of the metadata of the metagenomes. So from which location they are coming, from which depth layer they are coming here, and from which oxygen regime they are coming. So again, most of the pelagic ocean, we don't have some of the nitrospinas, but then even from metagenomes even from samples that they are not coming from our samples, we see this clustering, for instance, of the ancient, the greenish acid clades there. And the same with this group, another group that seems to be endemic in the in the dark green, which means anoxic metagenomes. A better representation is on a map. So instead of we lose the depth here because depth is not represented, everything is overlaid. But actually, the size of the circle is indicative of the relative abundance of them. So the way that works is this size is actually 4% of the relative abundance. And this group that we had shown in the tree to be actually exclusively found in the oxygen-depleted systems, we see exactly ETNP, ETSP, and the third oxygen minimum zones that we don't have salts from are Arabian Sea. Interestingly, when we do those distributions, two of the system, one of them is Cariaco Basin that Gordon has worked with, doesn't seem to have this this type of players, but it has some of the Nitro Maritima that has the widest the widest latitudinal range. But another system, like in the Black Sea, that seems to have, that is supposed to have very chemical similarities with Cariaco Basin, has another group. And I didn't have the time, and the same we see with these ancient clades. One of the ancient clades seem to be more prevalent on the North Pacific, and the other at the South Pacific. The thing that I didn't have the time to look into is what are the genomic features that dictate this distribution, but it is one of the next things that I will be looking at you, and then to revisit the origin of nitrospinas, because with the addition of these two ancient clades that we found from the from the actual anoxic water columns. What it has believed to be so far, the origin of them to have started in the normoxic ocean might not be true. So they might have originated from the anoxic or micro-oxic ocean. And then another question that I'm working with together with Natasha Gordon's group is how oxygen might affect the activity of these genes. Before going to the activity part, I also will want to briefly mention one of the other groups, functional group, the ones that we call like sulfide oxidizers. So another group that uses Rupisco as their gene to capture the inorganic carbon. So if you remember the percentages, they are the gamma proteobacteria. So all these orders that they are in purple are around 6%, and other phylums are 3 to 4, is around 3 to 2%. They are not very abundant, but as you see, sometimes in these oxygen-depleted systems, we have high numbers. And we have data from previous work that they might actually be very active there. As I mentioned, those have potential for sulfide oxidation. And I won't take a lot of time to go through like these three, but here with those colors again in these lineages are the different orders that like we recovered. We see the same separation that everything that is coming from the oxygen-depleted systems is clustering differently from things that they are from the normoxic ocean. They have also differences here. It's not like very obvious, but there are some circles there that they represent how big is their genome size and how big is the cell size actually. And there are some groups that they have like big cells and big genomes, and others that they are small ones. As I mentioned, like these are in the same like in the same genus, and they still seem to be distinct. And they also seem preference in depth. So the lighter color, the shallow the depth are, and there are some that they prefer the deeper ocean there. A thing that I want to conclude with for this part is that when I look on the different ways that they can use the sulfur compounds, and those are all these different the different genes in the periphery of the circle. Some of the groups that they are coming from the from oxygen-depleted system, they have a lot of them because there are a lot of sulfur compounds there. Whereas some of the other groups, they have only a couple of them. So it seems that this actually cluster differentiation of genomes is reflected in the genomic potential as well. And I mentioned like sulfur as a way to get energy. And here I have plotted other genes. Those are genes that the cells are using to respire oxygen. And those are genes that cells are using to respire nitrogen compounds. Again, when we have the genomes coming from these oxygen-depleted systems, you see that different genes for oxygen and nitrogen exist. But of course, when they are living only in the oxygenated ocean, they have only one type of terminal oxidases. I will summarize our results, saying these are not, I don't want to go into details, but for the sulfidic water columns, the black and the Baltic seas, we have a lot of more diversity of the chemoautotropes, but they are only a very small number of genes. So I won't go into details about them. But to summarize what we found in the open water oxygen deficient systems, we have these 5 groups that they are the most important ones. And the work that Natasha and Gordon are doing in their lab right now is trying to figure out which one, how much of this of each one of these groups is contributing to the carbon fixation. And this is not a trivial task, and it includes all of us working together. So, and that approach actually goes back to the dogma we have in biology that DNA, the genomic blueprint, is transcribed into RNA. RNA is only ephemeral, but it might give us like indication of activity. RNA is then translated into proteins, and then proteins actually determine how much of these gases or from this carbon is actually it is going on. So this is the process rate measurements. So we have done the first part by doing the identity and the functional potential. We are now working on this is work in progress to do the global distribution. We are finalizing the RNA part in order to be able to map RNA molecules back to DNA and see which of the pathways are active. And then what is happening in Gordon's lab is the last part. Actually, there are like several groups working on them. Collaborators from different institutions are doing the nitrogen cycling. But in all cases, how are we doing the rate measurements, which actually translates of how fast a process is occurring, is by giving a heavy, stable molecule that is the precursor of the process. We are feeding the microbial community with that. We wait for some time, and then we measure the products and we calculate rates. For the nitrogen cycling, we are mostly interested in the nitrogen compounds here. It's not as easy as it sounds. For the carbon is assimilated in biomass, and this is an example of a carbon, the stable carbon isotope. We can do that with radioactive as well. But since it incorporated into biomass, we can gather the biomass and measure how much it's going into the cells. And you're very privileged to have this the Raman microspectroscopy facility here, because this is what Natasha is using to be able to interrogate specific cells. So the way that Natasha and Gordon set their experiments is gathering and filtering the biomass there. I'm working together with Natasha in developing specific probes that they will target those individual groups that we talked around. And Natasha, Gordon, and their lab is working on interpreting the Raman signal in order to be able to calculate how much carbon is going into cells, and hopefully doubling times growth rates for those cells. We are interested in the big picture, so we want to put all those data in scenarios, in global models to predict scenarios of how it's going to look look into the future. And we have been in 2 cruises so far. One is coming. And I want to finish this talk by highlighting some of the new technology that we're developing. Instead of doing those incubations that I mentioned on board, we actually have small robotic systems that we send them at the depth that we want, and they do all this process in situ, because our problem with measuring those rates in anoxic systems is that we live in an oxygenated world, and only by sampling with the Niskin bottles we contaminate the process. And Natasha, in a few years, will present the progress of this work. The summary is, if you didn't hear anything, if you don't, if it was too much jargon, microbes are amazing. They have crazy types of metabolism, how they can transform one compound to another. They are by far the biggest chemists of this planet. But from our work, although we didn't recover a new per se group of these abundant players in the phylum level, we saw that in the fine scale, in the genus level, there are new players that we haven't been detected before. Both oxygen and depth, they are important ecological factors that they shape their distribution. Some of the lineages can use diverse energy sources, and I didn't have time to talk about that. But it's essential to link genomics with these ecophysiological studies in order to be able to have the right numbers that we need to plug them into the models. And I will finish by giving my kudos to all the people that they have contributed to this work and to my funding sources. This is my small but very very effective group. We have an incoming PhD student that will do the modeling part, and we're very happy about that. Gordon is one of the key collaborators from the carbon fixation side. Laura Bristow and Annie Bourbonnais are doing the nitrogen, the nitrogen part, and Jeannie actually introduced me into this world. The big data set is actually produced by Ramona Stepanauskas and what we call the GORDS Consortium. And with this, I'll take any questions. Thank you so much for your attention.
And so our first question goes to Taya.
Hi, great presentation. Thank you so much. I wanted to ask if you knew that when you're detecting these transcripts for Rubisco, if you're always detecting carbon fixation when you are seeing Rubisco in these microbes.
Yes, this is a good question, and we haven't, as Gordon knows more than me, much better than myself. Carbon fixation, especially with the stable isotope work, is very hard to detect because there is the natural variation that exists is just below B. So with the bicarbonate, when you use the radioisotope, you are much more have more related detection limits. A previous work that we have published and that happened in Cariaco Basin together with Mary and Gordon, we actually found a very interesting pattern. So when carbon is incorporated, it changes the fractionation. So it changes how it looks at the inside the cell. It discriminates for like isotopes. And literature says that different Rubisco forms have different fractionations, like fractionation efficiencies. And we actually were able to show evidence, and it was like a roughly, like a relatively small data set from the genomic side, but for the patterns that Mary and Gordon have seen only from the natural abundance of those of the profile, we were able to explain it based on how the Rubisco expression changed. So very hand-waving, I would say that the answer is that there is a correlation. One of the things that we try to develop, and my postdoc Julia is very interested in that, is by having control large volume intubation in order to be able, in the same water like incubation, do both the the rate measurements and the genomic analysis, because what happened that I couldn't go into detail, the way that it works, it is different. We sample from the same mini skin, but we do experiments in parallel, not at the same bottle, let's say. So we hope to have more concrete evidence that it's indeed like a closer connection of Rubisco and carbon fixation.
Thank you.
I will give it, yes. Go.
I was just curious, earlier when you talked about measuring the GC content, I just didn't know like what sort of motivations there were for that, if you were curious about resistant heat, stuff like that.
Yes, there are multiple, and that's a very interesting literature as well. Even how much nitrogen is in your environment can evolutionary change the ratio of this content. So for now, I'm going with an agnostic, so not expecting. I mean, not having an initial hypothesis why this might be different. So especially for the sulfide oxidizers that I see the highest differences in the GC contents between the different orders that they are there. I'll try to do some multiple inputs in those predictors that has to do with how many genes are there, what might be environmental factors that they are driving that, genome size, and other type of factors. But I don't have a good answer for that. One suspicion that I have is some of those groups actually might be mixotrophs. So if you can both fix inorganic compound when you have, but you can also eat organic matter if there is there, you might be able, because organic matter has carbon, but has also nitrogen. So that might be favoring your GC content one way or another. Great questions.
I was going to ask if anybody's ever looked at, we mentioned hydrogen as a possible
Yes.
chemical source, but there's not very much hydrogen data. So I just wondered if there's anything in the genes that tells anybody what's going on.
Yes. So there is a very it's a very there are genes that we call hydrogenases, but this is a very big group that has diverse functions. So hydrogen oxidizers, it's one of them. But cyanobacteria has hydrogenases. It can be both production and consumption. And on these cases, actually, there is a group, I don't remember the name, that has created an amazing tool that based on the phylogeny of the gene, you can infer its function, and it seems to work quite well. So I don't remember if it's a small, it's a small number, but there is indications that they are also hydrogen oxidizers there. Yes, not high numbers, small fraction of the community, and they might be using hydrogen plus alternative ways to fix carbon, to gain energy to fix carbon.
So I think you had an early slide that said both the nitrosospira and the nitrosplana had urease.
Oh, yes.
So I have an idea why an ammonia oxidizer might have a urease,
Yes.
but why do you think a nitrite oxidizer might have a urease?
Yes, yeah, that's a great question. And we have wondered about it for quite some time because we actually found that in the first, in my 2017 paper. So our initial hypothesis about that is that the majority of the ocean, like the ammonia oxidation is actually, or nitrite oxidation is coupled to ammonia oxidation. So if the ammonia oxidizers cannot turn, they don't produce nitrite oxidizers won't exist.
So they're helping out the ammonia oxidizers?
Michael Wagner's group, they have hypothesized also that, and they saw close proximity in wastewater treatments. In the ocean, it's very hard to say that something, it's symbiotic, but that was our initial. The fun fact that I didn't show this data is that it's not only there, like the urease is much more widespread than so sulfide oxidizers have it too, mostly in those like oxygen-depleted. So that's one of the things that I will be looking to, but especially for the nitrospinas, the entire cluster has been acquired from gamma protobacteria. So it's a horizontal gene transfer. They have that for a reason.
How big are their genomes? I mean, yeah, that's a big gene cluster. in
It's a
nature.
big class. The nitrospinas are on the big side, so they are around 2 mega or 2 point something. Yeah,
Still, that's not good.
yeah.
Thank you so much for this really interesting talk.